and welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Human Reproduction. Okay, and we are in part 5 of the series. So if you haven't watched part 1 to 4, please do so. All right, so let's begin. Okay, so question number 1. Match the following columns and select the correct option. Okay, and this appeared in NEET 2020. It's pretty recent. So you have column 1 and column 2 to match them. Let's match them then. Placenta, androgens, HCG, layer of the ovum, lubrication of the penis. So placenta secretes HCG. Okay. Zona pellucida is the layer of the ovum. You should know that by now. Bulbourethral glands, also known as Cowper's glands, secrete mucus inside the penis. So lubrication of the penis. And Leydig cells secrete androgens, yes, mainly testosterone. All right, so you have A2, okay, so A2, B3, C4 and D1. All right, so your answer is D. Pretty direct and simple, and complicated. Let's look at the discussion. Okay, so let's look at the discussion. Now, HCG is a glycoprotein. Okay, it's a hormone and it is a glycoprotein. Okay, and it is secreted by the syncytio or syncytio, whichever way you pronounce it, syncytio trophoblast cells of the placenta. Okay, so this hormone is very important. It is secreted by the syncytio trophoblast cells of the placenta. I hope you know what syncytio trophoblast cells are. So I have already spoken about this when I discussed implantation. So let's say you have your trophoblast cells, right? And inside what you have? Your inner cell mass, the embryonic stem cells, right? Now, once the zona pellucida, zona hatching happens, zona pellucida has disappeared, right? Now the job is for the trophoblast, the blastocyst, to be implanted into the endometrium cavity. Correct? That's the job. So now what happens is, now during that time, once that implantation has happened, or during the time of implantation, that's right before implantation, these trophoblast cells differentiate into cytotrophoblast cells and also give rise to another layer of cells called syncytio trophoblast cells. Okay, they also give rise to syncytio trophoblast cells. Now, syncytio trophoblast cells as such are not distinct from each other. They are, the cytoplasm is fused and there are lots of nucleus around. It's lots of nucleus around. Okay, syncytio trophoblast cells. And these trophoblast cells secrete HCG, and they also secrete many other hormones. We'll come to later on. We'll discuss the other hormones. And syncytio trophoblast cells also plays a very important role in making a connection, a blood connection between the mother and the fetus. Okay, so cavities form over here. Okay, cavities form over here through which maternal blood enters. Okay, maternal blood enters and leaves. Okay, so it forms a connection with the fetus. Okay, through, and we'll discuss that later, how placenta is developed. So chorionic villi forms there, and in between you have these intervillous spaces. Okay, where the maternal blood enters, and chorionic villi contains the fetal blood, and where exchange happens. Okay, so this is like the first step in creating that connection, the blood connection between mother and the fetus. All right, and from this syncytio trophoblast cells, many hormones are released. Okay, one of them is HCG, and that is human chorionic gonadotrophin. Now the functions are: it does not cause the involution of corpus luteum. thus maintaining the secretion of progesterone and estrogen. Okay, so HCG plays an important role. When we studied menstrual cycle, remember ovulation happens, right? If it is 28-day cycle, on the 14th day ovulation happens, right? After that is the secretory phase, which is dominated by progesterone, and of course estrogen is also secreted, and they are both these hormones are secreted by the corpus luteum, right? Now the secondary oocyte does not get fertilized, it will get discarded, right? Now, for example, if that does not happen and fertilization does happen, okay, fertilization does happen by the 7th day after fertilization, implantation happens, right? So by, let's say, 8th, 10th to 12th day, yeah, the implantation has happened and that bigerminal layer is also formed. Okay, now the syncytial trophoblasts begin to secrete HCG, okay, human chorionic gonadotropin. Now human chorionic gonadotropin plays a very important role in maintaining corpus luteum, okay, in maintaining that corpus luteum. It does not cause the involution of corpus luteum like it would do in a normal menstrual cycle or an ovarian cycle that we call it. Involution happens and then menstrual bleeding happens, right? But in this case we don't want the menstrual bleeding to happen, right? We want the endometrium cavity to be maintained because implantation has happened, and so now the baby is going to grow inside the uterine cavity, but obviously we don't want to lose the baby, right? So it is designed in such a way that HCG sends a signal to corpus luteum ki wait, don't disintegrate as yet, continue secreting progesterone, because progesterone is a hormone that maintains the endometrium cavity. It's only when progesterone is gone there's menstrual bleeding, right? But we want the progesterone to be maintained there by the corpus luteum. So continue, keep secreting that. So that is one reason. Okay, now this facilitates the formation of decidual cells in the endometrium, which become nutritious, containing glycogen and lipid deposits. Okay, so now progesterone also plays a very important role in causing decidual reaction. Now there's something called decidual reaction that happens in the endometrium. Okay, decidual reaction that happens in the endometrium. So what happens is progesterone, it creates an environment in the endometrium, in the uterus, to maintain the growth of the embryo. Okay, because embryo needs nutrition to keep growing, right? Only then it will form into a baby. All right, so the initial phase, this decidual reaction happens where decidual cells begin to grow over there, and they deposit glycogen and lipid, okay, for nutrition. Now Cl, that's corpus luteum, involutes in 13th to 17th week of gestation, followed by which placenta takes over its function. So by the 13th to the 17th week, placenta is fully developed. So now placenta is able to take over that. Okay, syncytial trophoblast cells begin to secrete that progesterone that is needed. Okay, now there's no job for Cl or corpus luteum to be maintained there. So that disintegrates, it involutes and is discarded. Okay, so now placenta is taking over. So corpus luteum is like a temporary endocrine gland that's present there. Obviously during the ovarian cycle it is present there, but even when the mother is pregnant, the female is pregnant, the corpus luteum is still maintained there in the ovary and is continuously secreting progesterone and estrogen till the placenta takes over. By the 13th to the 17th week, placenta formation happens. There's a connection established between fetus and the mother, where this exchange of blood through which nutrition is deposited, toxic wastes are removed from the fetal side, this exchange of carbon dioxide, oxygen, all that is happening, right? And that is then going on till the end, till the embryo grows into a fully developed fetus. Okay, so before that, let's come to before that. HCG maintains the corpus luteum, and corpus luteum then involutes once the placenta is developed. Okay, so in male fetus, and also one more thing, yeah. So now once placenta takes over and begins to secrete progesterone, HCG, there's no role of HCG, okay? HCG secretion stops. HCG is only responsible of sending a signal to corpus luteum, okay? So So in the beginning of the pregnancy only, you will find HCG secreted in the urine, or you'll find HCG in the blood. Okay, but before that, after that, once the placenta is developed, HCG will not be secreted. Okay, once placenta takes over and secretes progesterone to maintain the pregnancy. Just remember, progesterone is pregnancy hormone. Just think of it as this way. Okay, if you want to remember, is a pregnancy hormone that maintains the pregnancy throughout the gestation period. Okay, so first corpus luteum does it. Once the job is done, placenta has developed, the corpus luteum job is done, placenta takes over. Okay, so in that is one job of HCG. The other job is in male fetuses. HCG stimulates the production of testosterone from the testes till the time of birth. Okay, this is the second function. This is the first function. This is the second function of HCG. So in male fetuses, HCG stimulates the production of testosterone from testes, that is Leydig cells, till the time of birth. Okay, this causes the formation of male sex organ in male fetuses. Okay, so here what you see is the placental membrane. Okay, so here, like I told you, these are the uterine glands. Okay, this is where the decidual reaction happens. Okay, decidual reaction happens, and lipid and glycogen deposits are maintained here. Okay, so here syncytial trophoblast cells are here. Yeah, so here there used to be syncytial trophoblast cells before the cavity was formed. So now the cavity is formed, and now you see what? Now this insertion has happened of the arteries. Okay, so blood moves in. Okay, blood moves in, and also blood moves out also. Okay, so maternal blood is maintained here. Okay, this is maternal blood in intervillous space. Okay, now this is actually the chorionic villi. This is only one villi. Sorry, this is only one villi that we are seeing here. Okay, so I'll also discuss this later when I discuss the development of placenta. Or should I discuss this now? Let me discuss this now. Okay, so I'll discuss the development of placenta and the embryo. Okay, so we know till implantation, right? So in implantation, first before that, let me teach you the structure of the endometrial cavity. Okay, so this is the uterine cavity. Okay, this first part is the perimetrium. Then in between you have the myometrium, and then inside this part you have the endometrium. Okay, now the endometrium consists of 3 layers. Okay, what are those layers? The outermost is the compact layer. Okay, then the middle region is called the spongy layer. Okay, and the innermost layer is called the basal layer. Okay, it's called the basal layer, and eventually forms decidua basalis, and also called the decidual plate. All right, so this part is called the decidual plate. Eventually it will form the decidual plate. We'll come to that. Now, where does implantation take place? Now, implantation, let's say you have your cytotrophoblast here, your syncytial trophoblast have formed here, you have the inner cell mass here, right? Zona hatching has already happened. Now it's time to stick into that, right? So it will take place over here, okay? Somewhere over here between the spongy and the compact layer, okay? It will penetrate into the endometrium cavity and implant itself there. Now the cytotrophoblast cells and syncytial they are kind of sticky, okay? They are sticky, okay? And they hook onto the endometrial cavity, the layer, and then penetrate inside, okay? So they get implanted over here, okay? Now they get implanted between the compact layer and the spongy layer, and the basal layer will then form the decidual plate outside, okay? They form the decidual plate outside. All right, so and in the meantime what happens in the syncytial trophoblast cells? Cavities begin to form here, cavities begin to form here, okay? Cavities begin to form here for a reason. Why? Because once implantation happens, maternal blood will move in, okay? So now the decidua basalis contains that maternal blood, right? So there will be some penetration happening, okay? Some penetration happening. And for example, if this is your embryo now, okay, already the bigerminal layer is formed, okay, and syncytial trophoblast cells are here, okay, blood will enter, maternal blood will be present in these cavities, okay, which will eventually form the intervillous space, okay, once the chorionic villi has developed, okay. So this is the starting phase. Now this is what we have seen is the outside, okay. Now the inner cell mass is also differentiated by then by week 2 of development. Many things happen in the week 2 of development actually, okay. All that implantations happened within like the first week, right? Week 7, 8, 9, 10 days, around that time, okay. 7, 8th day implantation has happened. After that slowly the bigerminal layer will be formed, that is inside the embryo, the inner cell mass. Now what happens once that I've spoken about the implantation, now remember all this is happening simultaneously, okay. Syncytial trophoblast cells have formed, cytotrophoblast, all the differentiation has happened. Syncytial trophoblast cells form these small cavities inside, okay. And when they implant, okay, maternal blood begins to enter, okay, maternal blood begins to enter. So this is the first step towards a connection between the mother and the fetus, okay. It's the first step between the mother and the fetus, okay. Now this is happening on the outside. Let's talk about what's happening in the inside, okay. Okay, so now you have this embryo. Let's say there are cavities formed here, all right. You have the use a different color, you have the inner cell mass. Now this inner cell mass gets differentiated into two layers, okay. Inner cell mass gets differentiated into two layers and they are called epiblast cells, and second layer is called hypoblast cells, okay. Now the epiblast cells in particular are columnar, okay. The epiblast cells are columnar and the hypoblast cells are cuboidal, okay. And epiblast, please remember, epiblast is always on top, epiblast layer is always on top, hypoblast layer is at the bottom, okay. Epiblast layer is always on top, hypoblast layer is always on bottom. This whole region is called the embryonic disc, Okay, this is called the embryonic disc. Now think of this as a spherical structure. Okay, think of it as a spherical structure, because in embryology it is very, very important. We are 3D beings, right? Now from this, a human being, a fetus is going to get transformed, okay, from these two layers of cells. We're going to, it's going to evolve into a human being, right? So, and we are 3D beings, right? In the real world, in the natural world, we're all 3D, but what we are seeing here is 2D, right? But it doesn't happen like that as per the diagram. But you have to use a little bit of imagination. Think of it as a spherical ball. This is cytotrophoblast cells. Very, very important to understand. The cytotrophoblast cells are on the outside. Syncytiotrophoblast cells form there, right? And cavities form there. At the implantation, maternal blood begins to enter. Okay, maternal blood begins to enter. Okay, now the inner cell mass differentiates into epiblast and hypoblast. Okay, and this forms an embryonic disc. It's like a disc. Okay, it's like a disc, and you have your epiblast cells, and below that you have your hypoblast cells. Okay, so this is the bigerminal layer. Okay, this is called the bigerminal layer. All right, so now once this bigerminal layer is formed, later on in week 3 of development, this bigerminal layer will become trigerminal layer, but we will go into that later. First, I will discuss what happens with the outer section. Oh yes, and one more thing. There's one more thing that I wanted to mention. Now, when that bigerminal layer was formed, this you have the cytotrophoblast cells, there is syncytiotrophoblast cells there. You have your what do you call them? Epiblast cells here, and you have your hypoblast cells here. Now another cavity is formed here. So from the these trophoblast cells, another layer of cells appear called amniogenic cells. Okay, amniogenic cells. So this cavity inside, these are called amniogenic cells. Okay, these are called amniogenic cells, and the cavity inside is called amniotic cavity. Have you heard of amniotic cavity? I'm sure you've heard of amniotic cavity, but this is how it's formed. Okay, and this is also formed in week 2 of development. So once the epiblast cells, hypoblast cells are formed, another layer of cells also form on top over here. Okay, these are called amniogenic cells. Okay, and the cavity present inside is called amniotic cavity, which contains a fluid. Okay, we'll discuss that later. Now these hypoblast cells also give rise to another layer of cells which form the primary yolk sac. It doesn't contain any yolk, but it's called the primary yolk sac. Currently, for now, it's called the primary yolk sac. Later, it will develop into secondary, or it's also called definitive yolk sac, becomes smaller and more firm. Okay, for now, in week 2 of development, it is called the primary yolk sac. Okay, now this has happened after all that implantation has happened in the endometrium cavity. Now you know the 3 layers of the endometrium cavity, right? There's a compact layer, spongy, and basal, and implantation has happened. Okay, now obviously when there's implantation, I've already discussed this in the previous lecture, a hole like structure is formed. Okay, it's called a penetration defect, and that is covered with a fibrin plug later on. Okay, so now the endometrium is fixed. Okay, there's no defect anymore. All right, and at the same time, syncytiotrophoblast is what, secreting HCG, right? It is sending a signal to corpus luteum that please send in more progesterone. We need progesterone here, and decidual reaction happens. A decidual reaction happens over here in this layer. Okay, that means decidual cells form there, and there's more glycogen and lipid deposits. The cells become more epithelial, they become edematous, right? They become swollen with fluid in it, and they have glycogen and lipid to maintain the embryo. Okay, the embryo needs nutrition, needs energy to maintain itself. And also in textbooks they write that the decidual reaction is known as a defensive mechanism to protect the endometrium. Okay, because now there's a foreign body that has come in. Okay, the embryo is like a foreign body, and that has come in, and so now to maintain that endometrium, decidual reaction happens. And why, which hormone maintains the decidual reaction? Progesterone does that. Okay, progesterone does that. Basically, secretory phase in a normal ovarian cycle is usually applied to prepare the body for a fertilized ovum, right? But obviously that doesn't happen, so the oocyte is discarded. But if pregnancy does happen, fertilization does happen, and implantation does happen, that secretory phase becomes aggravated, it becomes more. Okay, it's more than usual. All right, so this is what happens. This is called a decidual reaction. Okay, so you have your amniotic cavity, you have your primary yolk sac. Okay, and amniogenic cells are present there. Amnio is iogenic cells. Okay, and you have a primary yolk sac. Now, this is week 2, but there is further development. Okay, now you have this embryo. Okay, primary embryo, that is, the initial stages of development. Now what happens outside? Okay, these cytotrophoblast cells secrete another mass of cells. Okay, they secrete another mass of cells and deposit them between this layer, primary yolk sac and amniotic cavity. Okay, the cytotrophoblast cells secrete another mass of cells and deposit them over here. That means the cytotrophoblast cells is pulled back, right? That whole layer is pulled back. Now if I have to draw another diagram, let me show you. Let me erase this. So you have your cytotrophoblast cells, right? You have your hypoblast cells. Yes, I'm sorry, your epiblast cells, your hypoblast cells. You have your amniotic cavity, and you have your primary yolk sac. Okay, I'm not drawn syncytial trophoblast cells, but another layer forms over here. Okay, so the cytotrophoblast cells give rise to another mass of cells that gets deposited between the embryo and that cytotrophoblast cells. Okay, now what is this layer called? This layer is called extra embryonic mesoderm. Okay, this layer is called extra embryonic mesoderm, this whole area here. So cytotrophoblast cells get pulled back, the embryo is there, right? And another mass of cells appear in between. Okay, so this is called extra embryonic mesoderm. Now what happens in the extra embryonic mesoderm? Small cavities begin to appear here. Okay, small cavities begin to appear in this region. Okay, and these cavities then become big. Okay, they begin to occupy the whole space. So if I have to draw the whole cavity here, okay, the small cavities begin to appear and they occupy that whole region. Okay, they begin to occupy the whole region. Now this division, okay, this whole cavity that has become superfluous, okay, it has occupied this whole region, gives rise to two layers. Okay, this gives rise to two layers. Here this region still contains the mass of extra embryonic mesoderm, sorry. So this is called the connecting stalk. Sorry, this is called the connecting stalk. Okay, and this eventually forms umbilical cord. Okay, this eventually forms umbilical cord, but we'll come to that later. Now this cavity that disappears and it becomes continuous, what is this region now called? This region is now called extra embryonic coelom. This is called extra embryonic coelom, which will later be characterized into chorionic cavity. Okay, which will be called chorionic cavity. And as you will see further, as the baby develops, as this bigerminal layer becomes trigerminal layer, the amniotic and the chorion begin to stick to each other. So the chorionic cavity also disappears. Okay, now as the baby grows, okay, it occupies a whole uterine cavity. This amnion area also, the embryo also is growing up, it forms into a proper body structure, right? And the chorionic cavity will eventually disappear. For now, in the initial stages, in week 2 of development, we have the bigerminal layer, we have this embryonic coelom that is formed, okay, and you have 2 layers formed in the extra embryonic mesoderm. Okay, so now the outer part is called somatopleuric layer or mesoderm, and the inner region is called splanchnic extra embryonic mesoderm layer. Sorry. Okay, so this is the inner layer, okay, and the outside region is called the somatopleuric layer. Okay, now think of it again as 3D. Okay, think of it as a burger. Okay, this looks like a burger. You have the patty in between, which is the epiblast and the hypoblast. On top of that, you have a balloon or a bun, which is the amniotic cavity. Below that, you have the primary yolk sac. Okay, now around that, you have the cytotrophoblast cells and the syncytiotrophoblast cells, right? They have implanted themselves. Okay, I'm just doing a recap. They have implanted themselves. The syncytiotrophoblast have formed cavities where the maternal blood is entering. Okay, this is the first stage or the first connection that is established between the fetus and the mother. Okay, now this cytotrophoblast cells give rise to another mass of cells. Okay, it's called the extra embryonic mesoderm. Small cavities begin to appear, which then again join. Okay, but not at the connecting stalk. Okay, the connecting stalk, there's no cavity there. That connecting stalk eventually forms the umbilical cord. Okay, now small cavities begin to appear, and because of that, 2 layers are formed. Okay, the outer layer is called somatopleuric extraembryonic mesoderm. Inside layer is called splanchnic extraembryonic mesoderm. Okay, now this splanchnic, if ever they ask you in the question, what is splanchnic extraembryonic mesoderm contain or cover? Okay, it covers the primary yolk sac. Okay, this region is the splanchnic extraembryonic mesoderm and covers the primary yolk sac, which is the amnion part. This part, the amnion part actually is covered by the somatopleuric mesoderm, extraembryonic mesoderm. Okay, the amnion part is covered by the amniogenic cells and the somatopleuric extraembryonic mesoderm. This splanchnic layer is mostly present in the around the primary yolk sac. Now, which is the chorionic layer or chorionic plate? Now, you know, this blue layer is called the cytotrophoblast cells. Beyond that, there are syncytiotrophoblast cells. Please remember, okay? Around this are syncytiotrophoblast cells with these cavities, okay, containing maternal blood. Okay, now another plate is formed, which is called the chorion plate. I remember I told you this extra embryonic coelom will eventually be called chorionic cavity. Think of it as this way, okay? This will be called the chorionic cavity, and the chorionic plate is very, very important. It contains somatopleuric layer, cytotrophoblast, and syncytiotrophoblast. Okay, so if ever they ever ask you what is present in chorionic plate, okay, this is very important. Chorionic plate will eventually give rise to the chorion, okay, chorionic villi, which forms the fetal connection with the mother in the placenta. Okay, So chorionic plate contains somatopleuric, extraembryonic mesoderm, then cytotrophoblast cells, cytotrophoblast, and a syncytiotrophoblast. Sorry. Okay. So this is called the chorionic region, and this is called the amnion region. This is splanchnic layer over here, okay, which is covered by the, as in primary yolk sac is covered by the splanchnic mesoderm, extraembryonic mesoderm. Now all this is happening in week 2. Okay, so a lot of development has happened in week 2. Inner cell mass has differentiated, extraembryonic mesoderm is formed, it has formed those 2 layers, extraembryonic coelom is formed, which will eventually become chorionic cavity. Now it's preparing itself to form the baby, okay, the fetus. All right, so this is all part of week 2 of development. I hope this is clear. Okay, so till now we have seen week 2 of development, right? Now let's look at placenta development in detail. Okay, placental development. So by now you understand the layers of the endometrial part of the uterus, right? Now let's look at what happens during placenta development. Now the embryo is fixed or is implanted in the compact and the spongy layer, right? So the decidua basalis, or the basal layer, it's also called the decidua basalis, forms the decidual plate. Okay, it forms the decidual plate. So this is called decidua basalis. Now why is it called decidua? Because at the time of parturition, this also slows off along with the fetus. So the whole placenta is removed. So decidua basalis is removed from the endometrium at the time of parturition. Okay, the time of process of giving birth to the baby, to the fetus. Okay, now we know that we have this cytotrophoblast cells here. I'm not going to embryo development in much detail. For now, I'm just going to talk about placenta development in this particular question. We'll come to that later, okay? You have your epiblast cells and you have your hypoblast cells, okay? And you have your amniotic cavity and you have your primary yolk sac, okay? Obviously by now chorion has developed, right? So let's talk about that in detail. So you have your chorion cavity. So let's say if this is your cytotrophoblast layer, okay, and outside that, let me use a different color, you have your syncytiotrophoblast cell. Now syncytiotrophoblast cells are very, very important. Let me not draw it like this. They all form a part of the what, chorion plate. Okay, so let's say if you have syncytiotrophoblast cells over here, okay, around this, imagine all of this in a 3D structure. Okay, imagine all of this in a 3D structure. So now inside this you have your intervillus space. Okay, now what happened is in syncytiotrophoblast, remember I told you that cavities are formed. I'm just drawing cavities at regular intervals. Now what do these cavities contain? They contain the maternal blood. Okay, that means arteries are entering, veins are leaving. Arteries sorry, and veins are leaving. Okay, now how does the chorionic villi develop? This is what I'm going to talk to you about. Okay, initially what happens is cytotrophoblast form these extensions inside the syncytial trophoblast. Okay, cytotrophoblast form these extensions in the syncytial trophoblast. Now, what is this stage called? This stage is called primary villi. Okay, this stage is called primary villi. All right. Now, once this is formed, what is the next step? These cytotrophoblast cells in the middle disappear. That means you saw this whole mass of cytotrophoblast cells. Now the middle mass has disappeared. Okay, the middle mass has disappeared. And you know this extra embryonic mesoderm is over here, correct? This extra embryonic mesoderm is over here. This develops over here. This extra embryonic mesoderm develops over here in between. This stage is called secondary villi. If they ever ask you, I don't know if they will ask you, but if they ever try to level up the game of NEET, they might ask you what is primary villi, what is secondary villi, what is tertiary villi. So there are 3 stages of chorionic villi development. There's primary, secondary, and there is tertiary. Primary mein what happened is cytotrophoblast cells, they invade that syncytial trophoblast cells. Okay, they form projections. Now the cells in between that projections disappear and they are covered with extra embryonic mesoderm, connective tissue. Okay, this is secondary. Now what happens in the tertiary villi? These connective tissue differentiate into endothelial cells. Okay, they form capillaries over here. Okay, they form capillaries over here. This is the third step, that means tertiary villi. Okay, they form these capillaries like structures over here. Okay, where now does it make sense? Exchange of gas will take place. Exchange of gases will take place. So from the umbilical cord, I have to use a different color here. Which color? Choose this. From the umbilical cord, arteries and veins enter. Okay, from the umbilical cord, umbilical arteries carry what? They carry deoxygenated blood from the fetus to the villi. Okay, and umbilical veins, they carry oxygenated blood. Now, maybe I'll talk about fetal circulation, or I'll make a separate video about fetal circulation. Okay, umbilical arteries, remember this. This could be a good MCQ question. Umbilical arteries, they carry what? Deoxygenated blood. Okay, and umbilical veins. Now, which is the other part? Which other artery or vein carries deoxygenated and oxygenated blood respectively? Put that in the comment section. Veins, they carry oxygenated blood. Okay, so umbilical arteries are carrying the deoxygenated blood from the fetus to the villi area. Okay, and then oxygenated blood is coming back into the fetus. Okay, so now there is a connection that is established. Okay, so you have all these capillaries also forming here, veins, venules, arterioles also forming here. Okay, of the fetus. Okay, differentiation of cells take place over here into endothelial cells. Okay, of the extraembryonic mesoderm. Now, if you can see, now you see a connection that's established between the fetal and the maternal. The syncytial trophoblast initially formed the first connection between embryo and the mother. Okay, Now this area is more developed. This whole area is called the placenta. This whole area is called the placenta. It's like a temporary organ that is formed to establish a connection between mother and fetus. Okay, so now blood enters, okay, and flows back. Okay, blood enters and then flows back. That's how this connection happen. Now, actually this villi space is kind of small, but let me show you. It's actually very well connected. Okay, they are very close to each other. These villi spaces are very close to each other. Okay, these villi spaces are very close to each other. I've shown you small boxes, but they are actually very close to each other. Okay, they become big later on. Initially, syncytial trophoblast cells, they contain very small cavities, but later on when the chorionic villi develops, they become big. Okay, and so what you see now is a space, an intervillous space. Now it's become an intervillous space. Initially they were syncytial trophoblast cavities. Now this space where the maternal blood is present is called intervillous space. Okay, now blood enters, okay, and then it flows back out. Blood enters and then flows back out. Okay, that's how this connection is established. Okay, so exchange of material takes place between this villi and this intervillous space. So arteries will bring in the maternal blood, exchange will take place, venules will take back that blood. Again, arteries will bring in the blood, exchange will take place, venules will take back the deoxygenated blood. Okay, similarly umbilical arteries are bringing in the carbon dioxide, okay, exchange taking place, and then it goes, then the oxygenated blood is coming back into the fetus. Okay, so again, once again, you have oxygenated blood of the maternal, okay, the exchange takes place between this intervillous space and the chorionic villi, and deoxygenated blood is going out. Okay, again exchange is taking place between the umbilical arteries and the veins, okay, to take back all that deoxygenated blood, all the toxic material, okay, and then bring in all the nutrition and all from the arteries. So arteries, they pump in blood with a lot of force. Okay, so it splashes down and then it goes back up, splashes down, exchange take place and goes back up, splashes down, goes back up. Okay, that's how this exchange is taking place, okay, between the chorionic villi and the intervillous space. That is all that's there to placenta. Okay, please understand what are the parts of the placenta and how it is developed. If they ever ask you what is chorionic plate, you know what chorionic plate is. You know that what consists of what are the placental barriers, you should know that also. Now eventually what I want to explain to you is, you saw this syncytial trophoblasm. So now as this begins to increase, the embryo begins to increase. Okay, now we are in the bigerminal layer. Already I've already shown you placental development, but all of it is happening simultaneously. Okay, so germinal layer becomes trigerminal layer. Okay, now trigerminal layer consists of what? Ectoderm, mesoderm and endoderm, right? Ectoderm, mesoderm and endoderm. Okay, via the process of gastrulation. This is happening inside the embryo. So what happens? Slowly the embryo is also growing up. After that, organogenesis happens. After gastrulation, organogenesis begins to happen and the embryo becomes bigger and bigger with time. Okay, now amnion cavity also expands, so because of which it begins to touch the outer region, the cytotrophoblast cells. So the chorionic cavity actually disappears later on, okay, as progression of the development happens, of the gestational period happens. Okay, the chorionic cavity disappears and this whole embryo juts out of the endometrial cavity. Okay, so if I have to show you a diagram, erase this. Okay, if the implantation has already happened over here, okay, implantation already happened, as it grows, this region expands. Okay, it expands like this. Okay, it becomes big. Okay, and eventually this whole uterine cavity is gone. The cavity is now filled with this embryo. Okay, the amniotic sac has become big. Even before that, what happens is the embryo folds and moves into the amniotic cavity. Okay, it folds and moves as the embryo is becoming bigger. Okay, after the trigeminal layer is formed, that whole embryo structure folds into the amniotic cavity, and where the rest of the development takes place. The amniotic cavity also becomes so big that this whole region is covered. The whole cavity is now consisting of the embryo, the amniotic cavity and the embryo. Okay, and the chorionic cavity of course disappears, surrounded by the trophoblast as a syncytial trophoblast cells. Now chorionic villi has formed with the placenta. Obviously, what I want to say is that the chorionic villi is formed around this whole layer, but with time there is less blood supply in this region. Okay, there is more blood supply in this region, so the chorionic villi is well developed over here. Now remember, I showed you those chorionic villi. They are called chorionic frondosum. Sorry. Okay, chorionic frondosum. Okay, in this part the villi degenerates. Okay, so it's called chorionic levi. Don't worry about all these terms, I just wanted to show you, let you know. Levi, I think, yeah, that's the spelling. Okay, so the part where the embryo is now, forming the placenta with the endometrial cavity, okay, with the decidua basalis, the decidual plate, is called chorionic frondosum. They form like finger like projections. Okay, they form finger like projections. And on the other hand, chorionic villi also develops there, but over a period of time, during the gestation period, that degenerates because there's not much blood supply there. So that area is called chorionic levi. That's all. Okay, so there is more villi in this region where the placenta is formed with the decidual plate, the decidual basalis region. Okay, so basically this is what I wanted to explain with respect to placental development. Okay, so yeah, so you have a cytotrophoblast cell. Some fibrous tissues are also formed here. So you have an outer syncytial trophoblast cell. Okay, and you have your syncytial trophoblast cell here. You have an intervillous space here containing maternal blood, and this is the villi over here. Okay, where exchange then now takes place. So maternal blood enters over here, and then exchange takes place and then leaves. Okay, maternal blood enters, leaves like that. Okay, there is continuous exchange happening between the mother and the fetus blood. Similarly, blood comes over here, the deoxygenated blood comes here, and then oxygenated blood goes back to the fetus. Again, same thing happens over here. All right, so this is it. This is what placenta development or formation is. Also, I wanted to add one more thing. After the tertiary villi is developed, it's nothing major, but the cytotrophoblast cells that were formed, see, this was surrounded, let's say, look at one villi, the cytotrophoblast cells, right, and the syncytial trophoblast surrounding it. Obviously there's villi around it. Okay, the cytotrophoblast cells form this very compact shelling like structure. Okay, so they pierce through this syncytial trophoblast cells and then form compact shelling. Okay, this is called cytotrophoblast shelling. That's all. Okay, that's all that is there. So now you have distinct intervillous spaces, you have distinct chorionic villi spaces where exchange of blood is taking place, exchange of nutrients, exchange of gases, all that is taking place between the fetal blood and the maternal blood. And that's the whole idea of placenta. That's called, now that area is called decidua basalis. The whole structure is called decidua plate over there because it slows off. Okay, it slows off at the end. All right, so this is what basically placenta is. All right, now for that you need to understand week 2 of development, to be able to understand how placenta is developed. Okay, so please go back and if you want, you can relook at my explanation again to understand this. Otherwise, you'll not understand placenta. Okay, if you don't understand week 2 development. All right. So if we go back to our question now, yeah, I clearly answered this. Sorry. Placenta syncytial trophoblast cells secrete HCG. Zona pellucida is a layer of the ovum. Bulbourethral glands lubrication of penis, both they secrete mucus substances, and Leydig cells secrete androgens. Okay, all right. So question number 2. Hormones secreted by the placenta to maintain pregnancy are, and this appeared in NEET 2018. A. HCG, HPL, progestogens, prolactin. Okay. HCG, HPL, estrogen, relaxin, oxytocin. HCG, HPL, progestogens, estrogens. HCG, progestogens, estrogens, and glucocorticoids. Okay, so hormones secreted by the placenta to maintain pregnancy are HCG, HPL, progestrogens, and estrogens. Okay, your answer is C. Okay, let's go back and let's go look at the discussion. So the hormones secreted by the placenta to maintain pregnancy are these. Okay, now let's look at the function. So estrogen is formed from substrates formed in the fetus. Now this is an interesting concept to understand, okay, because estrogen as such is not the precursors of estrogens are coming from the fetus, not from the mother. Okay, so pregnenolone, okay, very big words, is formed in the placenta from cholesterol. Okay, it enters the fetal circulation, and along with fetal pregnenolone, synthesized in the fetal liver is the substrate for the formation of DHEAS, that's dehydroepiandrosterone sulfate. So now what happens is pregnenolone is there, pregnenolone in the mother is there. There's some pregnenolone in the fetal blood also. Now that actually forms progesterone in the mother. Okay, it's the precursor for progesterone. But now this pregnenolone enters the fetal circulation also. Okay, and it forms DHEAS. Okay, and 16-OH DHEAS. Okay, it forms two substances. Don't worry about NEET. Okay, don't think of it in terms of exam, just understand the concept. Okay, DHEAS and 16-OH DHEAS is formed in the fetal adrenal, okay, adrenal cortex. Okay, this pregnenolone actually forms progesterone in the mother, but this pregnenolone enters the fetal blood also. Okay, that helps in the synthesis of DHEAS and 16-OH DHEAS from the adrenal cortex, the fetal adrenal cortex. Okay, now they are transported back into the placenta where DHEAS forms estrogen and 16-OH DHEAS forms estriol, which is the principal estrogen. Now there's an important interesting test that the doctors do. They check the level of estriol or the production of estriol in the urine. Okay, they'll test it if it's present or not. If it's present, that means they know that the fetal development is functioning normally. Okay, this is one of the tests that doctors tend to do. Okay, so DHEAS and 16-OH DHEAS, they go back into the placenta and estrogen and estriol is formed there. Okay, so now pregnenolone has entered the fetal blood and DHEAS and 16-OH DHEAS are formed. Now these go back into the placenta and form estrogen and estriol. All right. Yeah, so this is the diagram that I was talking about. See, pregnenolone goes into the fetal adrenal cortex, and forms DHEAS 16-OH DH, and that moves back into the placenta to form estradiol and estriol. All right. So this is it. So what are the functions of estrogen? Because enlargement, this is the important part to understand what does estrogen do. It causes enlargement of mother's uterus, enlargement of mother's breasts and growth ductal system for lactation, enlargement of mother's female external genitalia, and relaxes the pelvic ligaments for easier passage of fetus. Okay, so during parturition, it relaxes the pelvic ligaments for easier passage of the fetus. All right, so these are the functions of estrogen. The progesterone is derived from the pregnenolone in the placenta itself. Okay, it's not like estrogen where it goes into the fetus area and then does its job, and then DHEAs and OH go back into the placenta for estrogen formation. Okay, this is directly formed in the placenta. There's no problem at all. Obviously, understand when placenta is developed fully and progesterone, it begins to secrete progesterone, what happens? Corpus luteum involutes. Okay, it disintegrates. All right, so now we don't need corpus luteum. The job is done. What are the functions? Causes development of decidual cells for nutrition of early embryo. I've already discussed decidual reaction. Decreases contractility of uterus to prevent abortion. It is secreted by the fallopian tubes and uterus to provide nutrition to morula and blastocyst before implantation. Okay, helps estrogen develop a breast for lactation. So this helps estrogen. Estrogen does the main part in developing the breast for lactation, but this helps in the further development or more development of the lobules, the alveoli and the ductules, okay, for lactation in the breast. So these are the functions of progesterone. Now let's look at human placental lactogen, or it's also called as chorionic growth hormone prolactin, or human chorionic, such big words, somatomamotropin, HCH. Okay, previously it used to be called HPL, but now it's officially called as HCS. Okay, is a protein secreted from the placenta in the 5th week of pregnancy. All right, and secretion is directly proportional to the weight of the placenta. So as the placenta is developing, all right, this secretion will also increase. Now it is similar to growth hormone, and it appears that HPL, human growth hormone, and prolactin are derived from the same progenitor hormone. So it has the same structure as human growth hormone and prolactin. Okay, so it was also called as HPL, but now obviously the term is changed to HCH after they found that there are more functions to this. Let's look at the functions. It was known to promote breast enlargement, sometimes lactation, hence called HPL. That's why it was called HPL. Okay, causes formation of protein tissues like growth hormone, but it is weaker in action. It's not as strong as growth hormone. Okay, causes decreased insulin sensitivity and decreased utilization of glucose in mother, transferring it to fetus. Okay, so insulin sensitivity is decreased in the mother, and so that diverts the glucose to the fetus. Releases fatty acids from the fat stores in the mother as source of energy. Okay, now relaxin is also secreted and is released from the placenta and is a 48 amino acid polypeptide. It is known to soften the cervix at the time of delivery. Okay, so that's one of the functions that is known for relaxin. Sorry, let's go back to the question and see if you've answered this. Hormones secreted by the placenta to maintain pregnancy are, HCG, HPL, progestrogens and estrogens. Okay, so this is the appropriate answer. Alright, so let's move on. I think I've pretty much explained most of it. I explained placenta development, I explained the hormones related to placenta. Okay, so question number 3. The amnion of mammalian embryo is derived from, this appeared in NEET 2018. I've already discussed amnion when I discussed week 2 of development, and it is derived from ectoderm and mesoderm. Okay, answer is A. It's not endoderm, mesoderm and trophoblast, ectoderm and endoderm, no. So the floor of the amnion is formed by epiblast cells, okay, which eventually forms the ectoderm. So in gastrulation, we'll look at that later. Now the floor of the amnion consists of those epiblast cells there, and the lateral and the roof walls, okay, they consist of amniogenic cells derived from the trophoblast, and in the formation of extra embryonic mesoderm is surrounded by somatopleuric mesoderm. Now when we looked at amnion, okay, we looked at that amnion structure. This is your epiblast cells, okay, surrounded by this amniogenic layer of cells, okay, and it's surrounded by the somatopleuric layer, okay. So here you have your hypoblast cells, here you have your primary yolk sac, okay. So now these layers consist of amniogenic cells, but the outer layer consists of mesoderm, and then mesoderm is actually somatopleuric layer. Mesoderm, somatopleuric layer. Yeah, okay, so if you look at this diagram here, you see a secondary yolk sac is already developed here. You have your epiblast cells, you have your hypoblast cells over here. This is hypoblast, this is epiblast, this is amnion. Okay, this is amnion, this is the somatopleuric, this is splanchnic, this region over here. This is splanchnic. Okay, and this whole region outside is the somatopleuric extraembryonic mesoderm. So what you see here is the somatopleuric extraembryonic mesoderm is forming a part of the amnion. Okay, it's forming a part of the amnion. So that whole structure is the amnion, which will eventually become big in size as the embryo also develops and the embryo folds inside that. It will form a part of that structure. Okay, this is the future connecting stalk, which actually develops into umbilical cord. Okay, so think of it as a 3D structure. Okay, the umbilical cord moves there in, okay, and connects with the placenta, where the exchange of gases or exchange of blood is happening between the fetus and the mother. Clear? This was about amnion. Okay, so question number 4. Match column 1 with column 2 and select the correct option using the given codes. Okay, this appeared in NEET 2016. Mons pubis is female external genitalia. Antrum forms a part of the Graafian follicle. Yes. Trophoectoderm is part of the embryo formation. Nebenкern is mitochondria of the sperm, the fusion of mitochondria in sperm. Okay, so A3, B4, C1 and D2. So your answer should be B. So it is B. Let's go to the discussion. So mons pubis forms a part of the external female genitalia and is a fatty layer overlying the pubic symphysis. Okay, antrum is a fluid-filled cavity in Graafian follicle. Trophoectoderm assists in embryo formation. That's the trophoblast cells that are there around. You know what happens next, right? I've already explained that to you. Nebenкern is a mitochondrial formation seen in some insects such as Drosophila. It's not there in human sperm, but seen in insects. Okay, so after the formation of spermatids, the mitochondria of spermatids eventually form a massive structure called Nebenкern. Okay, so you have a diagram here. So this black region is called the Nebenкern. So basically it's the fusion of mitochondria and the spermatids in the case of Drosophila. Just remember it for that. All right, for NEET, if they ever ask. All right, so clear? This is all familiar now. Shouldn't be a problem, right? Let's move on. Okay, so question number 5. Several hormones like HCG, HPL, estrogen and progesterone are produced by, we just discussed this, is NEET 2016. It is placenta. Okay, answer B. Go back and watch my the discussion that I just did about the hormones in placenta. and I will not explain this further. Okay, these hormones are produced by placenta. Just learn it. All right. So let's move on. Okay, question number 6. Ectopic pregnancies are referred to as, and this appeared in 2015, A. Implantation of defective embryo in the uterus. B. Pregnancies terminated due to hormonal imbalance. C. Pregnancies with genetic abnormality. D. Implantation of embryo at site other than uterus. So, ectopic pregnancies are implantation of embryo at site other than the uterus. Okay, so the answer is D. So, ectopic pregnancy is when the fertilized ovum is implanted and develops outside the endometrial cavity. We've seen a normal implantation, right? So, the embryo moves into the endometrial cavity and stays there and begins to grow. That is normal pregnancy. But when there's an ectopic pregnancy is when that fertilized ovum is implanted anywhere else other than the endometrial cavity. So, then it becomes a problem. Okay, so types of ectopic pregnancies are tubal, ovarian, and abdominal. Okay, so it can occur in the fallopian tube, which is not right, occur in the ovary, which is again not right, or in the abdomen. Okay, so these are types of ectopic pregnancies. Just learn it as a definition. Okay, for NEET if they ever ask you. Okay, simple. Let's move on then. Okay, so question number 7. Select the correct option describing gonadotropin activity in the normal pregnant female. This appeared in 2014 and 2012. Alright, let's look at our options. A. High level of FSH and LH stimulates the thickening of endometrium. High level of FSH and LH facilitates implantation of the embryo. High level of HCG stimulates the synthesis of estrogen and progesterone. High level of HCG stimulates the thickening of endometrium. Okay. Now, high level of FSH and LH stimulates thickening of endometrium. No, that's incorrect. They do not facilitate implantation of the embryo. High level of HCG stimulates the synthesis of estrogen and progesterone. Yes. High level of HCG stimulates the thickening of endometrium. No, the progesterone stimulates the thickening of endometrium, not HCG. Okay, so your answer is C. So in a normal pregnant female, high levels of HCG stimulates corpus luteum to secrete progesterone and estrogen. High levels of progesterone stimulates a decidual reaction in the endometrium. I already discussed decidual, right? So let's look at this in detail. Now, what does decidual reaction include? Okay, transformation of connective tissue into epithelioid shape and are filled with glycogen and lipids. Okay, the endometrial glands dilate and increase in secretory activity. Blood vessels become tortuous matlab they become very twisted. Okay, and the stroma becomes edematous. Okay, they're filled with fluid. So this is what decidual reaction is. Okay, and it's a defense mechanism to protect the endometrium. So it's basically preparing the uterus for pregnancy. Okay, if they know that the implantation is done, HCG has sent a signal to corpus luteum, guys, I need your help. So progesterone, which is the main hormone, obviously estrogen also is involved, but progesterone is the main hormone which helps in this decidual reaction. Now, what does the decidual reaction include? All these things. All these events happen in that region. Okay, so this is what decidual reaction is mainly. Okay, so here I've already discussed now when I discussed placenta formation. So this is the endometrial cavity actually. Okay, so you have a compact, your spongy layer here, and this is the basal layer. This is then the muscle layer, the myometrium. So the basal layer eventually forms the decidua basalis. Okay, and this is where the implantation happens. Okay, and then the this way. It's this way. The chorionic villi develop over here. Okay, and this then further expands as the embryo develops further, okay, to become into a fetus, human fetus. All right, so I guess we can move on. I've answered the question. The answer is C clearly. The others don't match. Okay, question number 8. Which one of the following is not the function of placenta? And this appeared in NEET 2013. A. Facilitates removal of CO2 and waste material from embryo. Secretes oxytocin during parturition. Facilitates supply of oxygen nutrients to embryo. Secretes estrogen. Okay, so it does not secrete oxytocin during parturition. Okay, so the answer is B. So these are mainly the functions of placenta. Okay, you can go through it. Okay, note this down in your notebooks or whatever you use. Okay, it acts as a temporary organ and allows transport of oxygen, water, electrolyte. This will help you study for your exam. Okay, it eliminates CO2, urea. Maternal antibodies IgG pass through the barrier into the fetus. Placental barrier prevents many bacteria from reaching the fetus, but some viruses do enter. Okay, drugs taken by mother can pass through the placental barrier, like cocaine and heroin. If the mother is addicted to drugs, it can go past the placental barrier and into the fetus. Okay, antigenic reactions are prevented due to non-mixing of maternal and fetal blood. Okay, and placenta synthesis many hormones such as HCG, progesterone, estrogen, and HCS. Okay, so I would suggest you note this down because this is very important for NEET. Okay, this is very important for NEET. You need to know the functions of placenta. They can ask you anything. Okay, like we just saw. So now if you go back, yeah, they can disappear in NEET 2013. This is very, very important. All right, let's move on. Okay, question number 9. The first movements of the fetus and appearance of hair on its head are usually observed during which month of pregnancy? And this appeared in 2010. A, 4th month, 5th month, C, 6th month, and D, 3rd month. Okay, so the first movements of fetus and the appearance of hair on its head are usually observed in the 5th month. Okay, learn it as a fact. I have a table. Yeah, I have a table which I have taken from a book called Embryology. By the author I.B. Singh. Okay, so he has compiled a list of events. Okay, the development with time. So it begins over here. I put from 3rd month onwards. Learn this. Okay, for NEET, if they ever ask you, you know the answer. Okay, so you have 3rd month, you have the 4th month, you have the 5th month. Over here you can see the mother can now feel the movements of the fetus by 5th month. Okay, and hair on the head and eyebrows can be seen. Okay, so movement of the fetus and the appearance of hair are seen in the 5th month. You have 6th month, 7th month, 8th month, and 9th month. So if you want, you can note this down in your notebooks, although it's not there in NCERT. But if they ever ask you, you know the answer, right? Let me see which year it appeared. It appeared in 2010. Okay, so let's move on. Okay, so question number 10. Which extra embryonic membrane in humans prevents desiccation of the embryo inside the uterus? Okay, this appeared in 2008. A, yolk sac, B, amnion, C, chorion, and D, allantosis. Okay, so extra embryonic mesoderm, I already showed you. Extra embryonic membrane also, the amnion, the yolk sac, all that I have shown you, right? So and also the chorionic plate, right, the chorion cavity, all that I have shown you. So the membrane which prevents desiccation of the embryo is actually amnion. Okay, so the answer is B. Now let's look at amnion in detail. So amnion is a fetal membrane consisting of ectoderm layer of the floor and surrounded by amniogenic cells derived from the trophoblast cells on the lateral and roof wall. Okay, it appears in the second week of development and is a fluid-filled sac. So the amniotic cavity expands in the later development stage and covers the embryo and ensheaths the umbilical cord and the fetal part of placenta. Okay, so you see the amnion is all surrounded everywhere. Now the amnion consists of 2 layers, the outer somatopleuric extraembryonic mesoderm layer and the inner amniogenic cells. I already discussed this, right? So it consists of 2 layers. Now, the quantity of amniotic fluid is 1 liter at full term pregnancy. So, as the pregnancy develops, by the time the fetus is full term, the cavity consists of 1 liter of amniotic fluid. Okay, so in early pregnancy, it is colorless. Okay, the fluid is colorless, but towards the end of it, it is straw colored due to lanugo and epithelial cells of the fetus. So, lanugo is all the hair that is developed, okay, in the fetus and is removed. Okay, it's the hair that is removed. The water in the amniotic fluid is changed every 3 hours between the maternal blood. So it is refreshed. Okay, the amniotic fluid is refreshed. Now the amniotic fluid consists of water and solid. Okay, the solid consists of organic, inorganic, and suspended particles. Now the functions. It provides support to the delicate growing tissues of the embryo. It allows free movement of the fetus. So there is space between the fetus and the amniotic membrane. So the fetus has freedom to move around. Okay, and it protects it from injury and avoids the addition of fetus to the amnion. Okay, so the fluid is present there to prevent fetus from sticking to the amnion. Okay, and maintains an even temperature inside. Okay, maintains an even temperature inside. So it basically protects the embryo inside that cavity. Okay. And during labor, how does the amniotic cavity help? The amnion and chorion help in dilation of cervix. Okay, it guards against umbilical cord compression, and after first stage of labor, it flushes the birth canal by its bactericidal and aseptic antiseptic action. Okay, so yeah, so these are basically the functions. Sorry, these are basically the functions of amnion. Okay, these are basically the functions of amnion. Clear? We can move on. Okay, so question number 11. Which of the following hormones is not a secretion product of human placenta? Again, this appeared in 2004. You have A, HCG, B, prolactin, C, estrogen, and D, progesterone. Sorry. So, is not a secretion of human placenta, that is prolactin. Okay, so the answer is B. I think I already discussed prolactin, right? When I discussed endocrine system, please go and check that video there. This is the same information that I had put in endocrine system. Okay, so initially the prolactin inhibiting hormone that is secreted, okay, that inhibits the synthesis and secretion of prolactins. But when the mother, whenever she's pregnant, the concentration of prolactin increases from the 5th week of pregnancy until the birth of the baby. So this increases the amount of prolactin inside, and it is approximately 10 to 20 times the level in a non-pregnant female. Okay, the amount of prolactin. So after parturition, that means after the mother has given birth to the baby, the female has given birth to the baby, there's a loss of estrogen and progesterone. Okay, and that allows for secretion of milk by prolactin. So estrogen and progesterone some way were blocking that secretion of prolactin. So now you see this is a diagram over here. The blue line is estrogen. This is parturition, this black solid line here. This is parturition. Estrogen, after parturition, estrogen gone on, progesterone has gone down, sorry. And prolactin also goes down. Okay, it comes to normal levels. During pregnancy, it has gone to like 10 to 20 times the level of a non-pregnant female, right? So now but she's given birth. So it comes to normal levels after birth of the baby. But now each time the mother nurses the baby, the nervous signal sent to the hypothalamus causes surge of prolactin secretion which lasts for 1 hour. So prolactin secretion, understand that whenever the mother is nursing the baby during lactation period, it is not a continuous level. Okay, the secretion of prolactin is not continuous. There are surges. Okay, every 1 hour there'll be a surge. Okay, nervous signal is sent to the hypothalamus, which then allows for secretion of prolactin. It sends a signal to pituitary gland, and that secretes prolactin. Okay, which then produces milk. Okay, prolactin is responsible for producing milk, and this surge is lasts for every 1 hour. All right, clear? So now colostrum is kind of milk that is secreted first few days before parturition, last few days after parturition. Colostrum is very important for the baby. Okay, it contains the same concentration of lactose and proteins as milk but no fat. Okay, so in the next 1 to 7 days milk production begins in increased amounts, and along with that there is adequate secretion of other hormones such as growth hormone, insulin, PTH, cortisol for production. So now when the mother is producing milk, okay, now the milk contains all this, no, amino acids, fatty acids, calcium, glucose for the baby, right? Now baby is exclusively dependent on the breast milk, right, during the nursing period. Along with that prolactin is obviously secreted. Along with that there are other organs also that are secreting their hormones because they need to produce all this, right? It is all coming from the mother. The mother is the source, right? So she begins to produce amino acids, fatty acids, so insulin, growth hormone, PTH, cortisol, all that also begins to secrete. Okay, so the extra piece of information for you to understand where is all this coming from. Okay, the source is the mother, and so other hormones are also elevated during that period for milk formation. Okay, clear? I think I've answered this question, right? So it's not a secretion product of human placenta, that's prolactin. That answers our question, right? I've already explained what prolactin is in a bit of detail. Let's move on then. Okay, so question number 12. During embryonic development, the establishment of polarity along anterior, posterior, dorsal, ventral or medial lateral axis is called, this appeared in 2003, A. Organizer phenomena, B. Axis formation, C. Anamorphosis, and D. Pattern formation. Okay, so the establishment of polarity, that is the dorsal, the ventral, the left and right, the front and back, is called organizer phenomena. You will come across this when you study gastrulation. Okay, so the answer is A. Let's go to the discussion. So during embryonic development, the establishment of polarity is called organizer phenomenon. Okay, now there are different types of organizer: primary, the secondary, the tertiary. Okay, so the primary organizer is called the primitive streak and it induces the formation of notochord and intraembryonic mesoderm. The notochord determines the craniocaudal axis and right and left sides of the embryo. The notochord is a secondary organizer. The notochord becomes a secondary organizer and stimulates the development of the brain and spinal cord. Okay, and tertiary organizer is the neural tube, okay, which induces segmentation of paraxial mesoderm to somites. I know this is a little too much, information and doesn't make sense, right? So somites are usually, they are cells, okay, that form axial skeleton. They form the skeletal muscles and part of skin. Now I'll discuss gastrulation with you in this topic, in this particular question, because then it will make more sense. Now remember I spoke about embryo formation. The inner cell mass get differentiated into epiblast and hypoblast cells, okay, and obviously I spoke about extra embryonic mesoderm that's formed, and that whole placenta development, chorionic villi is formed. Fine, that is happening outside, but what is happening in the embryo? Okay, in embryo I told you that the bigerminal layer becomes trigerminal layer by 3rd week of development, and obviously the amniotic cavity also becomes big and the embryo folds in to the amniotic cavity. Okay, now how does this bigerminal layer become trigerminal layer? Now we are 3D beings, right? Think of it as in a 3D format. Okay, there's a burger. Okay, there's a amniotic cavity, there's a yolk sac, and there's a patty in between, which is a bigerminal layer. Now that becomes trigerminal layer. So let's say if you have this oval shaped structure, okay, this oval shaped structure over here, okay, this is the disc. Now what you're looking at is from the top, okay, this is the top of the disc. Okay, you have noto procaudal node over here, or plate it's called, procaudal plate is formed here, okay, the top. Okay, it contains buccopharyngeal membrane which forms eventually the oral cavity. Okay, so buccopharyngeal cavity forms here. Okay, membrane. And over here at the bottom, what do you have? The cloacal membrane, which forms the openings, the anal openings, the urogenital openings, the anal openings. Okay, now how does this, now what you're seeing is from the top. Okay, I want you to understand that this is the top view. Now what happens? How does this bigerminal layer become trigerminal layer? Now this whole formation, transformation of 2 layer to 3 layers called gastrulation. It has nothing to do with the stomach. Please don't assume that gastrulation, if they ever ask you the definition and if there is a definition related to stomach, that's incorrect. Okay, gastrulation has nothing to do with stomach. Okay, it is only the transformation of that. Think of it in embryology. Okay, it's transformation of bigerminal layer into trigerminal layer. Okay, that's gastrulation. Okay, so now what happens? Now a structure called primitive streak begins to appear over here. Okay, this primitive streak, this primitive streak begins to appear here. It's like a line, okay, that begins to appear here and ends in a node. Okay, there are cells inside. Okay, so this is called a primitive streak. It is similar to blastopore in animals. Remember when I spoke about gastrulation of frogs, there's a blastopore and from which all the 3 germinal layers were formed. Similarly, in humans, there's a primitive streak, and this is called primitive node. Okay, now currently you have on top, what do you have? You have the epiblast cells here, and below that you have the hypoblast cells. Right, now you have the procaudal plate, you have the buccopharyngeal membrane here, you have the cloacal membrane here. Right, I already explained the future result of the consequences of cloacal membrane and procaudal plate and buccopharyngeal membrane. Fine, now primitive streak appears from this cloacal side, okay, and ends in a node. Now what happens? Let's say if I had to cut, okay, if I had to cut over here, what do I see? Okay, now what do I see? Let's say you have your epiblast cells over here and you have your hypoblast cells here, and you have your layer here, right? You have your primitive streak here, you have a primitive node here, and you have your procaudal plate here. Fine. Okay, now what happens is there's a growth factor that is secreted from this primitive streak and primitive node. Okay, a growth factor is secreted from the primitive streak and primitive node. And what does that do? The growth factor attracts epiblast cells inside. Okay, before that, cells within the primitive streak and primitive node disappear. Okay, now this becomes hollow. Cells within the primitive streak and primitive node disappear. Okay, now what, before I said that there was a mass of cells here, but now the cells within the primitive node and primitive streak disappear. Okay, so now this part is not there. Now there's a dent over here. Okay, there's a dent over here because these cells are now gone. Okay, these, now this is now what is it called? Primitive pit. and this region is called primitive groove. Okay, these cells in between have now disappeared, and now that node area is called primitive pit. Think of it as a pit. Okay, it's hollow. Okay, the cells on top have gone. Okay, similarly in the primitive streak, the epiblast cells in that region have been eaten up. Okay, the epiblast cells have been eaten up. Okay, they have disintegrated. Okay, so now that has formed a groove. Okay, that has formed a groove and a pit. Now this secretes growth factors. Okay, this area secretes some growth factors. Okay, now this growth factor, what it does is it attracts epiblast cells into that pit and groove. Okay, it attracts epiblast cells from the pit and the groove. Okay, so now epiblast cells, now let's look at the groove part first. Okay, now cells begin to enter, epiblast cells begin to enter into the groove. Okay, and they move deep down. Okay, they move past the epiblast layer into the hypoblast layer, and they begin to displace the hypoblast layer. So now the hypoblast layer is gone, right? Epiblast layer displaces the hypoblast layer, and now what happens to the hypoblast layer? It's gone. Hypoblast layer is now converted into another layer of cells from the epiblast, and this is called endoderm. Okay, epiblast cells move into the groove. We'll talk about the groove first. Okay, epiblast cells move into the groove. They displace the hypoblast layer, and now these new cells occupy that space. Okay, and now that layer is now called endoderm. Okay, now that layer is called endoderm. Now this happens throughout. Okay, this happens throughout the disc. This happens throughout the disc. Okay, now what you're seeing is a top view. Okay, and what you're seeing now is a transverse section of that. Okay, if I had to make a cross section, what will happen? I will see now these epiblast cells have come inside and displaced the hypoblast cells, and it has occupied that area and has formed an endoderm layer underneath. Okay, so the endoderm layer is formed. Again, the next generation of epiblast cells enter. Once the endoderm layer is formed and is complete, another generation of epiblast cells enter into that groove. Okay, they enter into that groove and they form the mesoderm. They form the mesoderm layer. Okay, so now you see above the endoderm, you see another layer of cells called the mesoderm layer. Okay, again also they occupy that space, okay, between the ectoderm and the endoderm. Now ectoderm actually is the top layer, with the remaining epiblast cells form the ectoderm. That's what it is, but they form the middle layer. The yellow part forms the middle layer, okay, of this whole region, the whole disc. Okay, you have the endoderm in the bottom, which has been occupied by the epiblast cells. It has displaced the hypoblast cells. Then you have the next generation of epiblast cells moving into the groove, and they form the mesoderm. Okay, and the remaining cells then form the ectoderm. Okay, so this is how 3 germ layers are formed. Simple. It wasn't very difficult to understand, right? Gastrulation is pretty simple. Okay, so the groove is formed, and then the epiblast cells enter and form these 3 layers. They have formed the endoderm. Endoderm is formation is complete. They form the mesoderm, and the mesoderm formation is complete, and the remaining cells form the ectoderm, okay, of the epiblast cells. So that's how these 3 layers are formed. Okay, now one thing now that you see here, this embryonic disc from the top you see is an oval shaped. Okay, during the time of gastrulation it is oval shaped. Now as this endoderm, mesoderm and ectoderm layers are being formed, these cells begin to move up. Okay, it has to obviously occupy the whole area, right? Epiblast cells are entering to the groove. Okay, they're entering to the groove, and the whole embryonic disc is converted to 3 germinal layers, right? But by the time gastrulation is complete, by the time the 3 germinal layers have been formed, you will see that the embryo structure is not oval shaped anymore. It becomes pear shaped. Okay, the embryo becomes pear shaped. Okay, this was oval shape. Now this, think of it as this way. This is narrow, okay, and this top part is broad. Okay, this is the procaudal plate here, the cloacal membrane over here. Now if you think of it logically, you lost some cells here, you lost some cells here, but now these cells, epiblast cells, are moving and moving outside, moving into the groove and moving outside, right? So there are more number of cells over here, so obviously the top part will broaden up. Okay, there'll be one region where the top part, that will be the top part, that will be broadened up. Okay, so that's why you can pear shape. But the bottom part, this is the cloacal membrane area part, where the primitive streak starts, is narrow, but the rest of the region is broad. So that's why it looks like a pear shaped structure. Okay, it's not an oval shaped structure, it's not even anymore. Okay, now the primitive area part is a little narrow as compared to the broad area part, because cells are moving out laterally and upward. Okay, laterally and upward they move out, and the top area gets occupied more. That means there are more number of cells there, but obviously primitive node and primitive streak, there's been loss of cells over there, so that's why it appears narrow. Okay, so this is how that embryo changes, okay, in terms of size also. Okay, so previously it was a 2 germinal layer, now it's become 3 germinal layer with the primitive streak and groove, primitive streak and node, and it has formed a pear shaped embryo. Now you have seen 3 germinal layer have been formed, right? But now what about this node? We saw that there were cells lost over here in the node also, right? Okay, this area is also empty. Now what happens here? Here also epiblast cells enter. Okay, now when the cell, now this area is hollow, right? The epiblast cells have disintegrated that were there previously. It has become a pit, right? So now there is space here for more cells to come. Again, obviously growth factor is secreted over here also, and cells move in, epiblast cells move in, and notochord is formed. Notochord is formed. Okay, from here it goes up to the procaudal plate. Okay, notochord moves up to the procaudal plate. So if I have to cut a section over here, what I'll see is, I'll see ectoderm. Okay, this is the primitive node. Okay, and this is my procaudal plate. Okay, I'll see a tube-like structure coming out. It's a hollow tube-like structure coming out and going up till here, and this is my endoderm region. Okay, this is ectoderm, this is endoderm. Realize there is no mesoderm here. There is no mesoderm in the area of notochord, and this is a hollow structure initially. Okay, then after that disintegration of some cells happen over here. Okay, and it becomes and this part is exposed here. Okay, and then again it forms as a definite structure over here later on, and again this part fills in the gap. The endoderm cells and that notochord, definitive notochord, fills in the gap. Okay, so initially when the notochord is formed from the primitive node, it is hollow. Okay, then there is a disintegration of some cells between the bottom layer and the endoderm. Okay, and it quickly forms into a mass of cells, and that gap that was formed is then filled up. Okay, so this is a definitive notochord. Okay, initially it's a hollow notochord, then it becomes a definitive notochord. But realize one thing here, that there is no mesoderm. Okay, you have an ectoderm here, you have a endoderm, but there is no mesoderm. So which places in this embryonic disc don't have a mesoderm? Procaudal plate don't have a mesoderm. Okay, here what you see, if you take a cross section, if you look at it from a side angle, okay, you'll see that there's a procaudal plate here, there is a definitive, what is this? There's a definitive notochord here, There's a primitive node here, primitive streak, which then eventually disappear. Okay, so and there's a cloacal membrane over here. Okay, the buccopharyngeal membrane. So these regions are only surrounded by endoderm and ectoderm. Okay, these regions are surrounded by endoderm and ectoderm. So procaudal plate, notochord, and the cloacal membrane, they are fixed. The primitive streak, obviously there is now forming, there's a mesoderm forming there, but I'm only talking about cloacal membrane, the notochord, and the procaudal plate. Okay, these structures are tight together. Okay, they tight together, they don't allow the mesoderm from entering that region. The mesoderm will grow around it. Okay, so the regions in the embryonic disc where there is no mesoderm is procaudal plate, the notochord, and the cloacal membrane. Okay, so these regions don't have mesoderm. All right, so basically this is what gastrulation is, right, in brief. Well, this is what it is. There's nothing more to it. Yes, I've answered the question, right? During embryonic development, the establishment of polarity along anterior, posterior, dorsal ventral, medial, lateral axis called organizer phenomena. So now this gastrulation is completed. Now, when I spoke of paraxial mesoderm, I just wanted to say that from here, the intraembryonic mesoderm that is formed, okay, around this notochord, okay, that differentiates into different layers. Okay, there's an intermediate mesoderm, there's a paraxial mesoderm, and there's a lateral mesoderm. Okay, like this, it appears like this. This is a paraxial mesoderm, then there's an intermediate mesoderm, and then there's a lateral mesoderm. Okay, and then they further differentiate into different organs. Okay, so paraxial mesoderm forms into somites, okay, which then give rise to axial skeleton, the muscles, skeletal muscles, the parts of the skin, and so on. Okay, so basically this is the question is just trying to tell you, ask you that what organizes everything, which is the front, which is the back, which is the dorsal, which is the ventral. So gastrulation, I've explained how primitive streak plays a role. It begins the whole process and develops the notochord, and notochord with the brain and the oral cavity and the spinal cord will be formed, okay, in the top region. And the bottom region, the cloacal membrane region, you have the anal openings, the urogenital openings, which will again further, when you develop, the urogenital tract will be formed, and so on. So that develops further and further, then the gastrulation then leads to organogenesis, okay, where different organs are formed as the embryo develops into a fetus. Okay, so this is the beginning stage. Okay, this is the beginning stage. Till for NEET you need to understand gastrulation, you don't need to go into organogenesis, all that is MBBS. Okay, for now, for NEET you need to understand gastrulation. Clear? We can move on then. Alright, so question number 13. Extraembryonic membranes of the mammalian embryo are derived from trophoblast cells, B, inner cells, formative cells, and follicle cells. So the extraembryonic membranes are derived from trophoblast cells. So the mesoderm itself, it forms from the trophoblast cells, okay, and then it forms a cavity around, and then you have the amnion, you have the yolk sac and layers forming, and then you have the chorion plate also forming, which then develops into chorionic villi. Okay, so trophoblast cells play an important role in developing that mass of cells that form the extraembryonic mesoderm. From there, the other layers then begin to have a definite structure. Okay, so this appeared in 1994. Answer is A. So extraembryonic mass of cells is formed from cytotrophoblast cells surrounding the embryo. It begins to occupy the space between cytotrophoblast cells and yolk sac region. Small cavities begin to appear and become continuous till the sides of the amniotic cavity, forming a coelom in between and form two layers, somatopleuric and splanchnic. Okay, I already discussed this. Yeah, so this is same diagram. We've already discussed this, how the other layers are also from the amnion extra, with all these extra embryonic structures. Why they call extra embryonic structures? Obviously because they are outside the embryo. It's not in the end. Intraembryonic mesoderm and extraembryonic mesoderm are two different things, okay? So please don't confuse them. Intraembryonic mesoderm, I just discussed how that is divided further, but from the notochord area, but don't worry too much about that, but worry about extraembryonic mesoderm. You need to know extraembryonic mesoderm and how it is giving rise to definite structures of the amnion, the chorion, the chorionic plate. You need to know what it is and how that develops in placenta. Okay, clear? Let's move on. All right, so question number 14. Eye lens is formed from, and this appeared in 1992, ectoderm, mesoderm, endoderm, ectoderm and mesoderm. So eye lens is formed from ectoderm. Okay, so the answer is A. Learn it as a fact. I have a list of the different germ layers, and the fate of the germ layers. So what is formed? Okay, from ectoderm, mesoderm and endoderm. So learn it. All right. Eye lens is formed from ectoderm. So you see eye, the lens, retina and cornea are formed from the ectoderm. Okay. Gonads develop from embryonic, and this appeared in 1990. Gonads develop from embryonic mesoderm. Okay, answer is C. Yeah, gonads develop from the mesoderm. Okay, clear? Let's move on. Question number 16. Which of these is not an important component of initiation of parturition in humans? This happened in 2015, it's a cancelled exam. A. Release of oxytocin. B. Release of prolactin. Increase in estrogen progesterone ratio. Synthesis of prostaglandins. So is not an important component is release of prolactin. Okay, it is not an important component of initiation of parturition. The others are oxytocin, estrogen progesterone ratio changes and synthesis of prostaglandins. We will come to parturition in detail. So the answer is B. Okay, let's look at the discussion. Now when I was going through NCRT and I read the section on parturition, they have spoken about fetal ejection reflex, but they haven't really explained much about it. They just said that there's some well-developed placenta and the fetus, that they send signals and they allow for fetal ejection reflex to begin. Okay, that's when oxytocin is secreted and so on. But I thought that let me go through the whole parturition in detail from the start to the end for you to be able to understand what actually happens in parturition, but there are many factors going on at the same time. Okay, it's not just oxytocin, there are many other factors too. Okay, so let's see what is the starting phase. Now the fetal hypothalamus secretes corticotropin releasing hormone. Okay, and it is supported by increased secretion of maternal CRH. Now this particular phase is a hypothesis that researchers now assume that there is some signal that is sent from the fetal side, where CRH is released from the what triggers the release of CRH we don't know as yet, but there is CRH released from the fetal hypothalamus, and it is also supported by the release of increased secretion of maternal CRH. Okay, now once the CRH is released in the fetus, it stimulates the fetal pituitary gland to secrete ACTH. Okay, so the fetal side, you have CRH from the hypothalamus that secretes ACTH from the pituitary gland. Okay, now what does that do? What does ACTH do? It stimulates fetal adrenal glands. Okay, stimulate the fetal adrenal glands to secrete cortisol. Okay, it secretes cortisol. Now what does the cortisol do? It accelerates the maturation of respiratory system. Okay, it forms the final maturation of respiratory system in the fetus. So ACTH then signals the adrenal glands to secrete cortisol. Okay, now what does this cortisol do? It matures the respiratory system. Okay, now once this cortisol is secreted, it accelerates the secretion of placental estrogen and prostaglandins. Okay, so this cortisol sends a signal to the placenta, and the placenta begins to accelerate. Estrogen is obviously being secreted as normal. Okay, estrogen is being formed and synthesized there, but it increases the production of estrogen, and along with that prostaglandins also. Okay, prostaglandins also play an important role in the parturition process. Okay, it accelerates secretion of placental estrogen and prostaglandins. Now, what is the role of estrogen? We know estrogen is being accelerated, but what is the role? What does it really do? It increases the release of oxytocin from the maternal pituitary gland. Now, oxytocin is present in the posterior pituitary gland. It is secreted from there. Okay, so estrogen sends a signal to the pituitary gland. Okay, we need your help now. It's parturition time. All right, please send oxytocin from there. So pituitary gland begins to secrete oxytocin from the estrogen sends a signal to the pituitary gland, and pituitary gland begins to secrete oxytocin to the placenta region. Now it increases synthesis of oxytocin receptors in the myometrium, prostaglandin, and increase in gap junctions in myometrial cells. Okay, now the one of them is to secrete oxytocin, but it also increases the number of oxytocin receptors. Suddenly you'll find lots of oxytocin receptor cells in the body of the uterus. Another, it's and then the baby is there in the uterine cavity. Now it has to be pushed out, right? So the number of oxytocin receptors in the uterine cavity, the uterine body, begins to increase. Okay, so more oxytocin can bind to it, and more muscle contraction can happen and push the baby out, right? So oxytocin receptors begin to increase in the myometrium. The muscles layers are there, okay, where contraction is continuously happening. Okay, it also increases synthesis of prostaglandin and increase in gap junctions in the myometrium. Okay, there are some gap junctions which allow for signal transmission, okay, from one cell to another. Gap junctions are important, so there are increase in gap junctions also to allow faster signal to be transmitted, to be transferred. Okay, so there's continuous pushing and pressing of the myometrium, okay, of the uterus to push the baby out. Now it accelerates disintegration of amnion and decidual cells, resulting in the increased synthesis of prostaglandins. Okay, so obviously prostaglandins is secreted with the help of estrogen. How? This disintegration of amnion and the decidual cells. Okay, the amniotic cavity is there, the baby is inside that, the fetus is inside that. It allows for disintegration of these cells, and this results in increased synthesis of prostaglandins. It also stimulates synthesis of contractile proteins in myometrium. Now we need more actin and myosin to participate, okay, in this contraction. Okay, so it also increases the synthesis of contractile proteins in the myometrium. So estrogen plays a very important role in initiating all these processes. Okay, in this, it releases oxytocin, it helps in the synthesis of more receptors. Then there is prostaglandin synthesis, there's increase in gap junctions in the myometrial, there's more contractile proteins also. So estrogen is doing a very big role, playing a very big role in parturition. All right, it also increases excitability of myometrial cell membranes. Okay, so this is the role of estrogen. Now what is progesterone do? Progesterone really doesn't have much to do, because now it's time for the baby to come out. It doesn't need to hold the pregnancy anymore. Progesterone, what is progesterone? Is a pregnancy hormone. Okay, and so when there is decrease in progesterone, it means that it is not going to hold the pregnancy any longer. It's going to remove the fetus out. Okay, so progesterone levels begin to drop. The secretion of progesterone stabilizes or even slightly decreases at 7th month of pregnancy. Okay, whereas estrogen secretion from the placenta is continuous. So you see, placental estrogen is continuously increasing, but progesterone decreases or becomes almost stable, right? Estrogen is continuously being secreted throughout the pregnancy, but estrogen decreases slightly and then becomes stable by the 7th month of pregnancy, because now it's almost time, the baby is like almost developed, right? And now the progesterone doesn't really need to hold the pregnancy anymore in the endometrium. Now the fetal liver produces DHEAS. Remember when I spoke about estrogen formation synthesis, DHEAS is produced in the fetal, right? And it is transported into the placenta where estrogen estriol is produced. Now fetal liver produces DHEAS, a lot of DHEAS, okay, which converts to estrogen in the placenta. Now the presence of large amounts of DHEAS prevents production of pregnenolone and converted to progesterone. So basically it's like a negative feedback, okay? Now pregnenolone produced progesterone obviously, and it also went into the fetal blood to produce DHEAS, okay? And then DHEAS went into the maternal side to produce estrogen and estriol. But now what happens in parturition, there's too much of DHEAS going into the, there's too much of DHEAS going into the maternal side from the fetal liver side. There's too much of DHEAS and that's being converted into estrogen. Too much of production is already happening. Okay, so now they said now we don't need progesterone, we don't need pregnenolone. So pregnenolone stops, progesterone stops. Already DHEAS is already happening at a very high pace. So what happens? Estrogen has gone high, progesterone has gone down, right? Estrogen is increased, progesterone production has decreased. Okay, estrogen is up during parturition and progesterone levels drop. Progesterone levels drop. So that means the ratio is now changed, right? Previously during the pregnancy, progesterone was high. We needed more progesterone, yes, to maintain the pregnancy, right? We don't want to abort the baby, right? We need to maintain that pregnancy till it is fully developed. But now we don't need progesterone. Okay, progesterone also has a tendency to hold the myometrial muscles of the of the uterus, so that there is no abortion of the baby. Okay, so it maintains that pregnancy. But now we don't need it. So now progesterone levels have dropped and increased. There's an increased estrogen and progesterone ratio. So at the time of parturition, there is an increased ratio of estrogen to progesterone. I mean, estrogen is high, progesterone is low. Okay, progesterone levels are dropped, estrogen levels are high. Okay, so this is the role of estrogen and progesterone. What is the role of oxytocin now? It stimulates the number of oxytocin receptors increase in the myometrium and decidua, and increase in number becoming maximum at the time of labor. Okay, so the number of receptors basically increase, okay, at the time of parturition. So it allows for more oxytocin to bind. All right, it stimulates the release of prostaglandins from amnion and decidua. Along with estrogen, it also oxytocin also plays a role in secretion of prostaglandins. Okay, it causes uterine contraction. How? The stretching of cervix causes a neurogenic reflex which stimulates the paraventricular and supraoptic nuclei of the hypothalamus to increase synthesis of oxytocin. So as the baby is about to come down, cervix dilates. Okay, the cervix dilates. Okay, and this sends a neurogenic signal reflex to the hypothalamus. We need more oxytocin. So more oxytocin is sent, more contraction happens. Okay, more prostaglandins are being produced. Prostaglandins play a very important role in pushing, in causing that contraction to happen. Okay, even when you're having your menstrual cramp, also prostaglandins causing that contraction. Okay, so prostaglandins play a very important role in causing contraction, and that is stimulated by oxytocin. How? Dilation of cervix sends a signal to the hypothalamus. We need more oxytocin, so more oxytocin is being synthesized, and obviously the number of oxytocin receptors have also increased. That means it is very obvious that oxytocin plays a very important role in contraction of the uterus to push the baby out. Now, fetal pituitary gland also secrete oxytocin, which might play a role in exciting the uterus. Now, fetal pituitary gland, the function of fetal pituitary gland release of oxytocin is not known, but yes, oxytocin is present, but I would not give it too much thought to it. So don't worry about it. Just understand, just know for information that there is oxytocin secreted from the fetal pituitary gland. Okay, and also it might play a role in exciting the uterus, but this role is not established as yet. Okay, so don't give it much thought. Just think about the maternal oxytocin. The role of prostaglandins. It initiates and maintains labor and is synthesized in amnion, decidual cells, and myometrium. Its synthesis is triggered by rise in estrogen level, glucocorticoids, mechanical stretching in late pregnancy, and increase in cytokines. Okay, so there are many factors by which prostaglandin synthesis is increased. Prostaglandin increases the formation of gap junctions. I've already explained what gap junctions are. Alright, so these are the 3 roles of prostaglandin. Okay, now we have looked at estrogen, we've looked at progesterone, we looked at oxytocin, we've looked at prostaglandins. Okay, everything is just happening simultaneously. Everything is just being secreted at a higher rate, and their job is to push the fetus out. Okay, the job is to push the fetus out. Now, but this is the endocrine role. What I have just discussed is the endocrine role. But now there are mechanical factors also involved during parturition. Okay, there are mechanical factors also involved. Now, what are they? Now, the stretching of uterine muscles causes increase in contractility. So, every time the uterine muscles stretch, there is an increase in the contraction. Okay, the movement of fetus also causes increase in uterus contraction. So, fetus is moving out, it wants to come out. So, the movement of this fetus also increases on contractility. These are all mechanical factors that are involved. Apart from the endocrine, there are mechanical factors also. Okay. The stretching or irritation of cervix causes increase in uterus contraction. Okay, and there's a positive feedback which ensues. So, when stretching of cervix causes uterus contraction, it pushes the baby further down to stretch the cervix further. Okay, so now we have seen how oxytocin, prostaglandin, all those endocrine factors play a role, right? But now every time the cervix is dilating, the uterus is stretching out, the muscles are contracting. These are all mechanical factors, right? And the baby is being pushed down further and further and further with time. Okay, it's pushed down further and further with time. Now a positive feedback starts. Every time the baby is pushed out, the cervix dilates, there is more contraction happening. Okay, every time the baby pushes down and dilates the cervix, there is more contraction happening. Okay, so this is like a positive feedback. Okay, stretching of cervix causes uterus contraction. So cervix dilates, there is uterus contraction. Cervix dilates, is uterus contraction. Okay, this pushes the baby further down to stretch the cervix further. Okay, so every time a contraction happens, the baby will move down further. Again, the cervix will dilate. Then again, contraction will happen. Again, baby pushes down further, cervix dilates. Again, contraction will happen. So basically, this is what it is. Okay, so these are the mechanical factors during parturition. All right. Cervical stretching also causes release of oxytocin, and we know what oxytocin plays a role in the uterus contraction. Each feedback loop becomes stronger than the previous, resulting in expulsion of fetus. Okay. Now, neurogenic reflex in the spinal cord to abdominal muscles also cause contraction of these muscles. Okay, now we looked at uterus contraction only, but there are some neurogenic signals sent to the spinal cord from the spinal cord to the abdominal muscles, but they also cause contraction to push the baby out. So all of them are participating in pushing the baby out. There's endocrine changes happening in your body. There's mechanical factors around the uterus area where the myometrium is functioning now extra hard and extra strong to push the baby out. There are abdominal muscles also now contracting to push the baby out. Okay. Now these contractions assist in the expulsion of the fetus. Okay, so basically this is what parturition is. It's not just oxytocin release and also the mechanical factors that I just discussed, but there's more to it. There's estrogen, progesterone changes, and they've asked the ratio, so you should know what is actually happening with the ratio. right? So I would suggest you note these points down to help you understand, okay, better. There's oxytocin release, there's prostaglandin release, and there's an increase in estrogen and progesterone ratio. So estrogen increases and progesterone drops. Okay, so this is an important topic. I've already explained parturition just now. Note this down. This is very important for NEET. Okay, it's not just oxytocin. There are many other factors also playing a role. Okay. Obviously, prolactin does not participate in parturition, so that's not important. I've already explained prolactin also before. Okay. All right, let's move on. Okay, so question number 17. The fetal ejection reflex in humans triggers the release of, it's appear in Karnataka NEET 2013. A. Oxytocin from fetal pituitary, HCG from placenta, HPL from placenta, D. Oxytocin from maternal pituitary. So fetal ejection reflex in humans triggers the release of oxytocin from maternal pituitary. Remember I said fetal pituitary also oxytocin release, but that is not really established. It also helps in uterus contraction, but don't worry about it. Main is maternal pituitary. Always remember that. Okay, so it's the answer is D. Okay, and I've already explained parturition in detail, so I won't go back to that again. Fetal ejection reflex in humans triggers the release of oxytocin from maternal pituitary. Okay, and this is the role of oxytocin. All right, let's move on. Question number 18. Signals for parturition originate from, and this appeared in 2012. A. Both placenta as well as fully developed fetus. B. Oxytocin release from maternal pituitary, placenta only, fully developed fetus only. So signals for parturition originate from both placenta as well as fully developed fetus. Okay, so remember I spoke about parturition, the events leading to parturition. There's CRH from the fetus as well as from the maternal, and this results in a cascade of events. Okay, this results in a cascade of events. So when both are developed, a signal is secreted. Now the origination of that signal is not really known. Why CRH is being secreted, how does it get triggered, we don't know as yet. But yes, when we assume that when the fetus is fully developed and the placenta, they all ready now for parturition, the signal is triggered. Okay. Okay, so question number 19. Signals from fully developed fetus and placenta ultimately lead to parturition, which requires the release of, again, this is same question as previous, which requires the release of oxytocin from maternal pituitary. Okay, signals from fully developed fetus and placenta ultimately lead to parturition, which requires the release of oxytocin from maternal pituitary, which plays a very important role in uterus contraction. Okay, again, it's the same answer. Not discuss this further. All right, question number 20. I think all these questions are related to parturition. I've already explained parturition in complete detail. Now these questions have become very easy to answer, right? Fetal ejection reflex in human female is induced by, this appeared in 2009, release of oxytocin from pituitary, fully developed fetus and placenta, differentiation of mammary glands, pressure exerted by amniotic fluid. So fetal ejection reflex is induced by fully developed fetus and placenta, which then later on allows for oxytocin to be secreted. So the answer is B. So now, according to NCRT, parturition is induced by a complex neuroendocrine mechanism. Okay, this, and now you know what neuroendocrine mechanisms are happening, right? The signals for parturition originate from the fully developed fetus and the placenta, which induce mild uterine contractions called fetal ejection reflex. Now, this triggers the release of oxytocin from maternal pituitary. This is taken from NCRT. It is clearly stated here, and I've already explained what those complex neuroendocrine mechanisms are. Okay, and also according along with that, there are some mechanical factors also involved. All right, so I already explained that, and now you can answer this question pretty easily. I already answered this question, right? Yeah, let's move on. Alright, so question number 21. In human adult females, oxytocin disappeared in 2008. Stimulus pituitary to secrete vasopressin? No. Causes a strong uterine contraction during parturition? Yes. Secreted by anterior pituitary? No. Stimulates growth of mammary glands? No. So the answer is B. Okay, so oxytocin plays a role in causing strong uterine contractions during parturition. Okay. Yeah, I've already discussed this. Okay, so this is the role of oxytocin. I realize the last few questions are all related to parturition. They were all on the similar level. Okay, there was nothing really difficult asked. Okay, and I also gave you the definition of NCRT, and they said complex neuroendocrine mechanism. Now you know what those complex neuroendocrine mechanisms are, and it's not very complex. It's pretty simple. So I would suggest you go back to the parturition explanation and note this down in your notebooks, and it will help you in studying for your exams. Okay, right? All right. So I've answered the question. Okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today. And don't forget to like, share, and subscribe to MG NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for updates. And I'll see you in the next video lecture. Right? So bye.